All right, I'm going to go through the notes with you that covers the plasma membrane structure and function, and then we'll talk about some general transportation that happens throughout the plasma membrane, known as passive transport. So, here's my quote here. How did we get to know the cell membrane and its structure? So, the first thing was identifying which biomolecules or macromolecules are integral for the membrane structure, and that was the lipids and the proteins. And hopefully we remember the basic structure of lipids, but those are going to have the glycerol and fatty acid tails, and then our proteins, remember, are those amino acid chains that get folded in a variety of ways. So each of them will end up having their own, unique properties, and we'll talk about a few different types of proteins that are necessary for our cell membrane structure. But early on, the phospholipid bilayer was identified by using, here's our transmission electron microscope here. And we can see that, there's some sort of feature that goes around it. But there were scientists that were able to say, hey, like Robert Hooke was able to look at cork cells under the microscope and say, hey, they have this boxy nature to them. They clearly have something on the outside, but I don't really see that same boxy nature in our animal cells. But there's still something that protects animal cells. And then eventually scientists after that were able, to utilize this transmission electron microscope so they could actually see it. And they were able to say, yes, there's structure. And when the cell goes through and divides, it's going to create another spherical, there will be two spherical type cells in the end. So there's got to be something that's providing this spherical structure to our animal cells. And then furthermore, we were able to develop more technology where we could break down components and realize, hey, there's lipids in there, and there's proteins, and there's phosphates. Let's call this a phospholipid bilayer to describe the plasma membrane or the cell membrane. Okay? I'm going to refer to it as the plasma membrane from here on out. So phospho is referring to the phosphate, and then lipid is going to refer to the fatty acid tails that are there, and they identified that there's a bilayer. So there are two of them: phosphate heads, fatty acid tails, phosphate heads, fatty acid tails that link together. As this continued on, they were able to determine that it's the sandwich model. So that, like I was kind of identifying, these are my phosphate heads, and the lipid tails would go next to each other. So it was kind of like this sandwich happening. And then there were some proteins that would kind of build in there. They were able to identify, hey, there's multiple components to the membrane. It's not just phosphates, lipids, and proteins. There's many other features to it. So that's where the mosaic piece came into the model. They knew it was fluid and flexible, and if it were to puncture, it was able to regenerate and kind of heal itself a little bit. And it looked like proteins would kind of float within this molecule. So they call it the fluid mosaic molecule. It also had a name of the BLM, not Black Lives Matter, but it was a bimolecule membrane, bimolecule lipid membrane. Sorry. So that was the BLM, or a black lipid membrane that it used to be called. But for now, we call it the fluid mosaic model because the membrane can, it can bend, things can kind of touch it, and then it'll flex. So that's the fluidity. The mosaic part, again, if you're thinking mosaic in art classes, right, there's many different components to a mosaic piece of art. Same thing for this model. There are many different components. All right, moving on. So I was referring to our phospholipids, right? Here's our phosphate group, PO4. And so that's why it's the phosphate head. And then the lipids, remember, are made up of glycerols and fatty acids. So that's this component down here. And each portion is going to have kind of its separate feature to it. So phosphates up here are going to be hydrophilic. Remember that hydrophilic means they love water. So these are going to interact with our polar substances, okay? And then down here, we've got our fatty acid tails, fatty acids, lipids. These are hydrophobic, so they don't like water. So they're going to hang out towards the center of our bilayer here. And they'll interact well with, non-polar substances. So continuing with our lipid bilayer sheet over here at the bottom, once that kind of fades away, we'll see, that the polar heads are on the outside, so there can be water up here. There can be water down below because that's where the hydrophilic phosphate heads will interact. And then the fatty acid tails, right? They fear water. They don't like water, so they're going to hang out in the center of that and kind of touch each other that way because they are both the nonpolar features. And then again, you can see like if we're forming some type of sphere, right? It's going to take that same component and function piece where the phosphate heads are going to all be, there can be water and fluid in the center of the sphere. And then again, on the outside there can be more fluid. So those phosphate heads are there and then our tails are there. And that's the very basic feature that can be found in all types of cells. You'll also notice that our fatty acid tails typically are going to have, one saturated fat where it's the hydrocarbon chain, all single bonds. And then there also is typically going to be one unsaturated fatty acid tail where it has the double bond in there. So that causes the fatty acid to kink. And that's going to help maintain some fluidity between the bilayer here. So it has its fluid mosaic model. Okay. Continuing on, there's going to be many components to it. We just talked about those phospholipids and having saturated and unsaturated lipids that are linked together so that it can remain fluid. If it was completely saturated, then it would lose its ability to bend and flex. Cholesterol is going to be found within the cell as well. Cholesterol is going to be found within that phospholipid bilayer, and it's going to help keep them together so that when temperature changes exist, especially towards the higher temperature change, it doesn't just break apart. Proteins, there are many different types of proteins, but we'll look at integral proteins. So integral or integrate means it's going to go all the way through the membrane. And so we know that proteins can be folded in a variety of ways. So the top half that integrates with the phosphate, right, is going to be the polar portion of a protein. And then it'll have a nonpolar or hydrophilic area where it can interact with the tails. And these integral proteins can be channel proteins, they can be transport proteins, and they're just going to allow for certain substances to flow in and out of the cell. So those are the integral proteins. And then there are also peripheral proteins, which are going to attach to the top surface of the membrane, and those are going to help with, again, maintaining structure. They're going to allow for attachment of specific molecules. And again, if they're peripheral proteins and they're at the surface of the membrane, these are going to be polar so that they interact with our hydrophilic phosphate heads. So continuing with the mosaic piece, right? There's lipids, there's proteins, there's cholesterol. There are also going to be carbohydrates, and I like to call these carbohydrates the communication centers for the cells. And this can include some glycoproteins and glycolipids to help with cell communication and cell recognition, but they kind of look like little trees that stick out of the cell. And they wave their branches in the air, and eventually something will come link up to those carbohydrates, and then communication will happen within the cell, and the carbohydrate will say, hey, I have this sodium ion right here. Is it needed, or should I keep it outside? Is it dangerous? Etc. And then if it is something that is needed, that the carbohydrate will ship it on over to one of those integral proteins so that it can pass it through the cell. And if it's not good, it'll kick it out or make sure that it's dissolved in some manner. So there's lots of communication happening inside and outside of the cell for transportation, which is something we'll talk about in a little bit. So here's our fluid mosaic model, again, kind of shifting with how it can flow and bend. We talked about, the bilayer already. So we see the phosphate heads and the fatty acid tails and how they're straight and kinked and all that. Here are our cholesterol, right? We don't want tons of cholesterol, right? Your doctor says that's bad. And it is bad if we have too much of it. Then this fluid, it would lose its fluidity, and it would be really, really too strong, too tight. So that would be dangerous. And then, sorry, that's our cholesterol. And then we have our proteins. Here is an integral protein, or one that looks like a channel. So it's going to be tubular. So, something can travel through it and get across the membrane if it's not small enough. So we see those integral proteins, and this is an example of how its protein folding can impact its general shape. And then we have some surface proteins or peripheral proteins here and over here. And then, there's a couple up here. And those are going to help maintain structure. And then our last thing, those carbohydrates, kind of like trees with their branches hanging around up here, right? So they're going to be sticking out. And here's what is known as a glycoprotein. So that's going to provide, the solid base for those carbohydrates to hook onto and then hang out and then communicate with what's going on in the cell. So we see all sorts of different features. And again, we got the outside of the cell up here in the extracellular fluid, and this would be the inside with the cytoplasm. We can start to see that there's other features with the cytoskeleton and stuff like that. So lots of components for the cell membrane. And yes, you do need to know each of them, and you should be able to identify where they are. Remember, kind of structure of what a carbohydrate looks like. So that's why we see kind of the ring shapes that are attached to each other. All right, continuing on. The cell membrane is known as a fluid mosaic model, but it's also known to be selectively permeable, meaning that it will select what can exit and enter the cell. Right? We don't want anything to just freely flow in and out of the cell. It could be something that harms it. It could be something that's helpful, but we also want to be able to regulate how much is entering and exiting, and making sure that the good stuff goes in and the waste gets kicked out. So that's why it's selectively permeable. And so we had those proteins on the previous slide, those integral proteins that are larger, and they will provide transportation for large or charged molecules that can't quite just fit between our phospholipid bilayer, whereas others will be able to just squeeze through those little membrane pores. So again, here's those channels. So large charged molecules can flow in and out. Otherwise, there's going to be little spaces between these phospholipids where, like oxygen, carbon dioxide, water, all those things can free flow between the inside and the outside of this cell. Okay. So continuing with that transportation, we're going to talk about passive transport and what